design at ATREE in the United States. The title of the talk is Ecological Underpinnings of Robust One Health.
Hello everyone, my name is Dr. Vanak. I am the Director for the Center of Policy Design. Apologies for not being there in person, but I hope this virtual version of me works just fine. I'm available for questions after this talk. So today I'm going to be talking about the ecology as the basis for robust One Health systems. So what is One Health? I'm sure you'll hear this term quite a lot. One Health, of course, as defined by the CDC, is a collaborative, multi-sectoral and transdisciplinary approach to working at the local, regional, national level with the goal of achieving optimal health outcomes. In particular, it recognizes the interconnection between people, animals, plants, and their shared environment. Put very simply, One Health means that if you want to have healthy people, you also need to have healthy animals and a healthy planet. So given this, it sounds very, very clear that this intersectionality that they're talking about is actually what people like me who study ecology call ecology. So it's the study of interactions. And how do we put this in the context of disease systems? So if you think of any kind of infectious disease, we typically think of infectious diseases having some sort of spillover, some sort of transmission mechanism. This transmission mechanism could either be from human to human single species transmission, or it could be a cross species transmission, in which case it's often called a zoonotic spillover, which involves animal to human transmission. Typically, how does a transmission of this sort happen? So let's examine some ecological factors or land use change factors that might result in these kinds of transmissions, and what does it mean for us when we want to bring ecology into the very basis of this. So let's imagine that there is some reservoir host distribution. Say, for example, bats. They could be bats, they could be rodents, they could be any kind of wild animal, primates, and they live in some natural space. There is also a natural infection, a viral infection and shedding cycle going on. Again, it could be, I'm saying viral, but it could be any pathogen, bacteria, whatever you want to replace them. And if there is some pathogen shedding going on, then you also assume that there will be some recipient hosts that are available for exposure. And you might, if this happens, then you assume that there will be a spillover. In the case of zoonotic diseases that transmit from wild animals to humans, sometimes is also a bridging host, which usually is a pet animal or a domestic animal. What we have done through human-induced changes to landscapes is that we have allowed for the interactions between wild animals and our domestic animals or directly ourselves to increase. So therefore, the probability that a wild animal that is normally in its natural range will then come into contact with a domestic animal host increases, especially during this time of viral shedding and infection. Let's also consider that when you make these changes, you're also inducing extra stress into those animals, into those wild animals, and therefore increasing the chance that they're shedding, they're infectious and they're shedding. Okay? That their cycles have gone beyond a certain allostatic load, and therefore the chances that these animals can then shed their pathogens to humans increases, and this increases with either clearing natural areas for agriculture, clearing natural areas for building cities, or for increasing areas for farming, for animal farming, whatever it is, any kind of landscape change that and only by reversing some of these are you able to reduce the potential for any kind of spillover to happen. Now, countries like India, of course, we don't have this luxury of separating where people live and where wildlife are. In this map, I want to highlight a couple of things. If you see all the red areas in this, you will see that places like India are very bright, which means that areas that have the highest livestock, areas with the highest human population, and areas with the highest biodiversity overlap. And India, in this bottom map here, this map shows the risk of emerging and re-emerging infectious disease. And you can see that India and Southeast Asia, South China, etc., are really amongst the hottest of hotspots. So they're at the highest risk of emerging and re-emerging infectious disease. And this is because countries like ours have a highly saturated interface between people, domestic animals, and areas of biodiversity. In addition, when people access livelihoods, they often tend to access livelihoods from natural areas where wild animals live, and therefore they're increasingly exposed to zoonotic disease. So in this talk, I will give one example of work that we're doing to deal with Kyasanur forest disease, which is a vector. So how do you optimize benefits that people obtain from these natural areas while still minimizing impacts of diseases like Kyasanur forest? And how can these lessons also be extended to others? So we know that there are many diseases that are borne by ticks, and ticks are a major carrier of several types of viruses. Say, for example, in Europe and North America, we have Lyme disease, which is a major public concern. In India, you have diseases like Kyasanur forest. And this was first discovered in Karnataka, in areas around Shimoga in Shimoga district. This is a flavivirus that causes, at its end stage, fairly debilitating hemorrhage. Initially, it used to result in greater than 500 cases per annum, but the cases have come down over time, and the mortality has also. Mainly people from forest-dwelling community, forest workers, plantation workers, were typically the most affected. And this disease has a very complex transmission cycle, which includes multiple species of animals, including ticks, rodents, small mammals, primates, birds, and other animals found. Several ticks have been implicated in the transmission of this as transmission vector. And there are several things that we still don't know about the contribution of various small mammals and other animals in the transmission of this disease. And we also don't know what factors, human factors, affect the risk of contracting this. So we shall examine some of these. Basically, the tick itself goes through several stages, and at any of these stages, it could get, especially if it's taking a blood meal, it could become host to the virus. And at the next stage, when it matures to the next stage, it then seeks another blood meal where it can then potentially infect another animal. It could be a small mammal like a monkey, or it could be a small mammal like a rodent or a shrew, or a medium-sized mammal like a monkey. Now, monkeys are known to be sentinel species for this disease, and that's why it's common name monkey fever. Because once you see monkeys dying in the forest, then it was assumed that this fever is prevalent, or this disease is prevalent, and people were cautioned to not venture into the forest. Now, of course, this is not possible because tens of thousands of people are dependent on the forest system for their livelihood, and this disease is now found in several states, potentially affecting millions of people. These are the typical landscapes that this disease is found in. So these are, for example, in Shimoga, in Tirthahalli. So typical landscape with paddy in the valleys and plantation, areca forests of secondary forests, primary forests. So a really interesting mixed landscape. So in this landscape, what we did is a interdisciplinary OneHealth surveillance, where we used ecological sampling techniques to look for ticks on the forest floor and other vegetation. We also sampled livestock, because livestock can be carriers of or amplifiers of ticks themselves, even though livestock are not known to be amplifiers of the virus in particular, but they can certainly be amplifiers of tick population. And then we did a series of, we captured lots of small mammals to look at which hosts are likely to have infections of Kyasanur forest disease. Then we also looked at the behavior of people. We wanted to know which habitats people went through when they were walking through their normal, when they were going through their normal daily routine, and where they were more susceptible to catch ticks, because obviously ticks are the primary vector. So we wanted to know where the ticks are present, and by seeing where they were going, we would be able to then advise them to use the appropriate prevention measures for preventing tick infestation. So why are all these methods necessary? This is because most models and decision support tools for zoonotic diseases tend to only focus on single set of processes that drive risk. For example, they could only be social, they could only be behavioral, or they could only be ecological or epidemiological. Instead, if we bring these three together, we are able to better focus on environmental hazards, and incorporate social processes that drive exposure. So by doing this, using this OneHealth system, we can also then link the intervention programs to scales that are appropriate to the sources of epidemics, and the ecosystem. So for example here, we map the hazards. The hazards are the ecological community, the rodents, the ticks, the monkeys and other wildlife. And we looked at the exposure. So the exposure is the resource use and priorities of the forest. And only by combining this then you can get a disease risk. So we asked how do these social and ecological risk factors vary and how are these linked to forest structure across the forested landscape. So once we took all of these parameters and then we input them into some model, including and then built predictive models that allowed us to then test which areas are most susceptible for outbreaks of KFD. So we took so all the black spots in these models that you see are from past cases of KFD. And so we use these models to match patterns in KFD cases at these village, household, climate, forest landscapes and hosts. And based on this, we are able to predict where KFD would occur in future. So based on our models, and I apologize for the sound disturbance, based on these models, we were able to predict KFD risk in advance of emergence and spread. And these models are now used by the health department, the Department of Health and Family Welfare in Karnataka, and there is a decision support tool that's been made available to the DHFW so that they can input data as it comes and they can help showcase their IFCAI in this kind of work. Obviously requires a very diverse team of people. This team included ecologists, sociologists, epidemiologists, public health workers, modelers, veterinarians, and a whole host of institutions, both government and private, that allowed us to do this kind of interdisciplinary one. So it really requires ecology coming together, ecology as being the basis that for all of this, underpinning all, but also social sciences and epidemiological sciences and public health sciences to come together that allowed us to then understand risk factors for KFD that we can then present to the government, make a decision support and help prevent these. In future, we need to carry out these kinds of exercises for almost all of our zoonotics and for other factors as well. That really builds on this network of different sectors and brings them together, allows for communication, collaboration, coordination and capacity and a broad stakeholder. And only in this way are we able to bring One Health or use the concept of One Health to really build a strong framework for preventing these kinds of infectious diseases from. I'm happy to take questions. There are many such different groups coming together. For example, this is the Bangalore One Health City cluster, and I encourage the audiences here to think of how more clusters such can be formed. I look forward to questions from you. Thank you.
Have you looked at the role of wild animals in the spread of Kyasanur forest law?
Yeah, am I audible?
Yeah.
Hello.
I hope my question could reach you.
Yes. So the study did in fact looked at the role of wild animals, and so the sampling that I described earlier consisted of extensive sampling of small mammals, especially things like forest-dwelling rodents, shrews, and so on. In the transmission dynamics of this virus, and we looked at the kinds of ticks that were found on these and whether they were infected with KFTB or not. So wild animals are the primary reservoirs of KFTB and play a very important role in the circulation of this virus and the emergence of this virus into human communities.
I have the ethics committee of one of the studies from Manipal University. It's a state from where I come from, where they run a project on Kyasanur forest disease as well. And they've been trying to correlate the paw marks of tigers and the ticks found and association with KFD, because they've been looking at the virus in the ticks collected from the areas where these tigers were spotted. So that was the reason for my quest. Thank you.
That is an excellent talk. Basically, the langurs that are susceptible for this KFD virus, not the other bonnet macaques. Other macaques are New World monkeys, so they are not the hosts, and they, so far, whatever research work that has been gone in for a long time, there is no wildlife associated KFD disease. Not even the reservoir have been identified. Maybe it requires a lot of sampling and other things. So that is one thing, observation. And actually it has a cycle of 7 years. If you go through the KFD, once in 7 years, it goes for a outbreak like situation. Epidemic peaks come up, and not only Shimoga and South Canara, North Canara are affected, it goes up to Chitradurga. Also, you do get 1 or 2 cases coming up. This is what the work I also had worked when I was in Government of India, and this is epidemiological studies that we did in those days. We have the data with us. Thank you, sir.
Yeah, thank you. Respectfully, I will disagree with some of those observations, sir. Basically, the work that we've been doing in the last few years very categorically shows that a whole host of small mammals, so wildlife includes rodents, includes, for example, the Blanford's rat, several species of shrews. In fact, AFTV has been found in a whole host of birds, bats, rodents, etc. And including monkeys. The bonnet macaques are indeed old world monkeys. Bonnet macaques, in fact, have been found dead in most of these, including langurs. Langurs, of course, there, but bonnet macaques have also been found dead and to be affected by high viremia from this virus. These are all results that have come recently, the last few years, from Nivedi, from NIV and other institutions. Our work is very conclusively showing that the primary reservoir, very large community of wildlife hosts, which, as I said, includes small mammals. So it's important for us to focus on the prevention aspects. The vaccine currently is not available. New vaccine is under development. But how do we prevent people from getting the virus? And this is the only way to do so is by ensuring that the ticks, that they are not susceptible to ticks. And earlier it was thought that only in a 50-meter zone of a dead monkey becomes a hot zone. But we have found that this is not the case, that the virus itself is prevalent across a very wide area, and people are susceptible anywhere. So our main intervention really is to prevent people from getting tick infestation.
Thank you.
Thank you.